Alright everyone, welcome back my fellow Mountain Shadow scientists. Today in the lab, we are going to be extracting DNA from strawberries. Now DNA exists in all life's organisms. It's called deoxyribonucleic acid. It's a small microscopic, you have to see it in a microscope to see, and it controls everything that happens in our body. All of our bodies have cells that have DNA in it, and that DNA is the building block of life. Now, in order to extract the DNA, we are going to need a couple things. But first, let's go over some fun facts about DNA. DNA exists in all living things, from humans to animals to trees to the fruit that we eat, the vegetables. Everything has DNA in it. And when those DNAs collect together, it's called the genome. Now, in order to extract DNA from strawberries, we are going to need in your kit some rubbing alcohol that's been put in the freezer, and that's really important. Is you want your rubbing alcohol to be nice and chilled, some salt, some dish soap, some coffee filters, 2 cups, a plastic baggie, and our strawberries. Now the reason why we're using strawberries is because most cells only contain 1 copy of DNA in them, whereas strawberries have 8. So when we're using strawberries, there's more DNA material to work with, and thus we can see it better when we extract it. In order to extract DNA, you have to have a solution, which we use for the detergent. Now the detergent helps the cells kind of pop open when the DNA is released by the rubbing alcohol, which is the catalyst of the extraction. And then the salt is what helps that DNA, especially strawberries because there's so much more, it helps that DNA find each other and bind together in the dish soap solution. Now, most labs that extract DNA use a very similar process, although they have much more sophisticated equipment than we do, but it's the exact same process that they go through. So, the first thing that you're going to do is you're going to take some strawberries and you're going to put them in a Ziploc bag. You want to make sure that the air is out and the bag is nice and sealed. You also want to make sure that the stems have been cut off as well. So you're going to take your strawberries and you're just going to start smashing them with your palm. And this is to help release the DNA from inside the strawberry. You know, all the juice, all those microscopic cells that are bound together inside the actual strawberry. You want to do this for a couple minutes, you know, no more than a minute and a half, 2 minutes. So there we have our mushed strawberries where the DNA is just waiting for us to extract. So we'll put that to the side for a minute, and now we have to create our extraction solution. You're going to want a cup of water, about half a cup of water, and you're going to add a couple teaspoons of salt. Remember that salt helps bind those DNA cells together in a clump. And then you're going to add about a tablespoon of detergent, and that detergent helps pop open those DNA cells, and then the salt collects it. So once you have that solution together, you're going to mix. Get that mixture nice and the salt dissolved. You don't want to create too many bubbles, so you just want to make sure that you're just doing it gently. Alright, now we have our solution. So we're going to take our strawberries again, and we're going to pour some of that solution into the bag with the strawberries, like so. Alright, then we're going to seal the bag up again, make sure that the air is all out. Alright, and then we're just going to smash our solution into our strawberries. Make sure that they're nice and mixed together. That salt and detergent are starting to bind together those DNA cells so that when we add our rubbing alcohol, the solution will come right out. So here's our mixture. Now we want to take our other cup, our empty cup, and we want to take our coffee filter. This is going to take some finesse to it. You might need 2 coffee filters, but you'll put that coffee filter down into the cup, and you will pour your solution down in there. You want to try to make sure that none of those strawberry chunks get into the mixture. You can pull it out so you see it's draining into the cup. You just want the liquid. You can gently squeeze it. Just make sure that none of the chunks come out. Okay. You can keep going until you've gotten it all out, but for time's sake, we'll just leave that be. Now, here's the important part. You have your chilled, stick in the freezer for at least an hour before you perform this. Now, you don't want to just dump the rubbing alcohol in so that it, just mixes all up. Right now it's got some bubbles in it, so those bubbles would need to settle before you do this. But you just want to gently pour the solution in, and what that's going to do, it's going to help extract that DNA from the strawberry, and then the salt and the dish soap are going to help bind them together, release that DNA. And hopefully, when you do that, and the reaction will take a couple minutes for you to actually see. You don't need to pour all of it in, just enough where you have, twice as much as the liquid that was already in there. So there we go. So now what's happening inside the cup is that the DNA is starting to slowly be extracted. You can't see it yet, but inside the cup, the DNA is starting to be pulled out from the juice by the rubbing alcohol. And what you get, now it doesn't look like much right now, but that substance at the top of the lid is actually the DNA. It looks a little bubbly because that's what the detergent was to release that DNA into something with the salt so that it coalesces together. And you can see that it's like this gooey substance right there. And that is actually the DNA of a strawberry. How cool is that? So have fun with your experiment of extracting DNA. We don't need a microscope to see this because strawberries are awesome and they provide us with 8 copies of their cell just for this purpose. So have fun, enjoy, and come back next time for another fun Mountain Shadow Science project. Make sure to subscribe.